So on Monday, we started talking about research methods, and this is apparently where we're going to get a little bit behind. I knew we'd have to finish up today, but we're still in recording neural activity, and we still need to go through the structure of the living human brain. And I'm backing up just a tiny bit to go back over positron emission tomography, or the PET. And I'm not going to go over the whole thing. Just what I think is important to take away from this is that we do use radioactively tagged chemicals. They're introduced and serve as markers of blood flow or the metabolic activity of the brain. So they're telling us where the blood is flowing and where the neurons are active, telling us about brain activity. And we do get a color-coded map of the areas where the brain is active. And we stopped here with the Peterson et al. Study from 1988, now a really classic study, where they had subjects perform several different tasks, and we're comparing looking at words to when they're listening to words to when they're saying words out loud, either read or heard, or when they are thinking of words. Okay, so in this case, a word association task. If they see apple, they might think about orange. Okay, so when they're looking at words, we're looking at the left hemisphere, so the front of the brain is to your left. So this is the back of the brain. When they're just looking at words, we see activity in the occipital lobe, which makes sense for visual processing. And as we are listening to words, you can see that the activity here is in the temporal lobes, the auditory cortex, probably here to near Wernicke's area as well, as we're listening to words, some of those auditory association areas. When we're speaking words, we see more activity there at what looks like probably right around Broca's area as they are saying the words, that motor area for when we speak. And when they are thinking about words or thinking about a word associations, there really is activity over a great deal of the brain. I'm going to say this with words on a slide, but just so you can see, we get this map, this color-coded map of where activity is, but you can see that it's also it does not have good spatial resolution. We don't have a good indication of exactly where that activity is. It's just kind of this general area, and I'm making those assumptions about where that is. It's been a long time since I've read this paper, so based on what I'm assuming is going on with looking at, listening, and speaking, but it's difficult to tell. I'm going to use the example of listening to words. It's difficult to tell where exactly in the temporal lobes that is. And I started with that because PET has some drawbacks. One is that it's slow. Okay, experiments typically take 2 hours. As we're looking at that metabolic activity, and we need that radioactive chemical to decay, that each condition and each trial takes some amount of time. So they are usually pretty slow experiments. There's this low resolution, not just spatially as I was showing, but temporally. Again, we need that metabolic activity, takes time, and then it's expensive. There needs to be a nearby source of radiation, and it says there are risks associated with a radioactive tracer, and that's not really true. I mean, it's extremely small and it's not really any risk, but people don't like to hear that they're going to be injected with a radioactive tracer, and so people would sometimes balk and say, I don't think I want to do this after they had already volunteered. So as most of us know, the functional magnetic resonance imaging has really taken the place of PET a great deal. It is much more common these days. If you notice, that PET study was conducted back in 1988, that was quite a while ago. And when we were just starting to figure out how to use this MRI machine to look at the functionality, to look at the brain activity. The magnetic resonance imager itself is using magnetic fields, radio waves, and computerized enhancement to map out brain structures. And we'll talk about this more when we're talking about looking at the structure of a living human brain. we use this variation of the MRI. The functional MRI is measuring blood flow and oxygen consumption in the brain to show us where we have high activity. It's sometimes called BOLD, so blood oxygen level dependent imaging. And so what's happening is hemoglobin, our blood, has a ferrous component to it. So it lines up in a certain way in the magnet, like little tiny magnets within that. Those people are surrounded by a magnetic field. And then when the neurons use the oxygen because they're active, that hemoglobin lines up differently. And the machine can tell us where those differences are to say this is where the brain is active. So as I said, the fMRI is preferred to PET. Like PET scans, we can map brain activity over time, but with vastly greater spatial resolution. So within 1 to 2 millimeters, you're going to see some pictures and you're going to see that they are much more precise in where we're talking about where brain activity is. And greater temporal resolution as well, so greater resolution in time. And I'm not sure exactly how long the PET takes for a kind of trial or task, but within about a second, we're going to see that metabolic activity. And as I was saying earlier, for EEG, or really the ERP, the event-related potentials using EEG, as well as the magnetoencephalography, the MEG, that remember those were giving us the temporal resolution of milliseconds. And so milliseconds are a thousandth of a second. And for me, somebody studying language, that's really important. I'm speaking really quickly here, and you're responding to my words within milliseconds. We usually spend, I think the average is something like 250 to 300 milliseconds on a word when we're reading. So a second isn't going to tell me about necessarily natural processing. And language isn't the only example. There are lots of tasks and things that we process really quite quickly. And but this gives us within about a second, which is better than the PET scans. So I'm showing an fMRI machine here, or an MRI. It's all the same thing, DTI. It's the same machine. And I usually ask people to share their experiences here, and I get some interesting stories. And so if you want to share on Monday, please think about doing that. I'll probably forget and not ask, but if you do, that would be great. I'm going to share my experience since I'm the only one here talking today. So I went in for a research study for a friend of mine, and it was a pretty terrible task for me because I do have ADHD and I do have a hard time inhibiting a wrong response. Oftentimes I don't inhibit that wrong response. This task was a Stroop-like task, but instead of colors and color names, there was a line, and you had to say whether or not the word was above or below the line. And sometimes those were just words, regular words, but sometimes the word, let's say above, that word was below the line. So I'm seeing the word above, but I have to say below. And it was very hard for me. Sometimes I have a student in the class who really goes on about how horrible these machines are. Every once in a while, if you have any kind of claustrophobia and you're going into the machine, it's very uncomfortable. And if you have severe claustrophobia, of course, some people just cannot do it. But I didn't find it that bad. And if you ever get a chance to run in a study where they're looking at fMRI or diffusion tensor imaging, it is a really good experience to have that perspective of the technology. I hope some of you will share your experiences on Monday. So I'm doing this task. If you're in a research study, you're doing this task, and then every once in a while, and as far as I could tell, there was no predictability to it in time, but they would have this bang, 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 click, 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 bang, bang, bang. And it wasn't even that predictable. Sometimes the bangs and the clickas would change numbers. It was as far as I remember. And of course, they did find that pituitary adenoma, that little cyst on my pituitary gland, as I was doing this study. And so in the study, they had something strapped to my face where they were presenting the stimuli. So I couldn't really even move my head without these images moving with me. It was very, that was disorienting to me. But going in and getting my cyst monitored every few years that doesn't really bother me at all. But sometimes people are pretty claustrophobic and they have a really hard time with the machine. So I'm showing these areas of the brain, and again, you can see on the top in the top in A, that the red is showing you activity, and the bottom is the B is the yellow. They usually do either red or yellow. I think it's usually yellow is the highest activity, and then red, then orange and red, and then the green and blue is the low activity. And you can see how precise, and especially if you kind of go back and imagine that PET study of what we were looking at, this is much more precise, and we can look at these really small brain regions. This picture is showing the parahippocampal place area that is activated by the top row, so those places, but not to the bottom row next to it. So jet planes, jumbles, and things do not activate it, but anything even suggesting a place. So on the far right, those to me look like Legos or something that are suggesting potentially navigating through here. The one next to that is an empty corner of a room. So yeah, even anything suggesting a place activates this area. And then on the bottom in B is showing the extrastriate body area. If you look at the top row, that's what activates it, and it's any part of the body, a whole body, even a stick figure or a shadow of what looks like a human figure. But the bottom row, all of that, even the face, right? Even though we can think of that, of course, it's part of the body, but we have that fusiform face area that is processing faces, and so that doesn't belong in the extrastriate body area. But again, what I'm showing here is the precision of where that activity is. So I'm going through most of this I said with other slides, but here it is with words on a slide. We can do those studies that are similar to PET experiments. Subjects can do almost any task. The advantages are this shorter time window, the reasonably good images of the brain, and we're studying activity in a human intact brain. The disadvantages are that loud, annoying environment, and even though it's faster than PET, we're still not capturing the millisecond by millisecond, that kind of level of processing, that kind of temporal resolution, which we talked about EEG and MEG earlier. They can do, but they're not giving us this precision of exactly where, if you remember, where in the brain that activity is coming from, because we're summing over thousands and thousands of neurons. And your author addresses the subtraction logic, I think here, after both PET and fMRI, and I'm going to use his pictures as well. But you know, we've gone over this for PET, that we are going to have an experimental task, but the brain is always active. So we really want to see what's the activity that's due to this specific task, and so we have to have some kind of comparison task, and we're going to subtract that comparison task, the brain activity to the comparison task, from the brain activity to the experimental task. I think this example was looking at words was the experimental task, although I'm pretty sure the example's made up, but he gives us an example of a possible subtraction. So my experimental task might be looking at words on a screen and reading. And then my comparison task might be something like looking at a foreign language that we know the person doesn't understand. So they're looking at something that's very word-like. The visual, what's going on visually is similar, but the component of reading has been taken out. And what I'm assuming he's really showing there in the difference with the +5 and really the +2 behind it is it's those left areas that are near Wernicke's. He looks like he's showing Broca's area, but we have Broca's area active often when we are even reading words in that this embodiment idea of what part, even though we're not moving our lips and making those motor movements, our motor area is active. So we see that activity in the left, but you can see along the left hemisphere, we're going to see that activity in those language processing areas. and we're subtracting out the comparison task, so we're not really seeing the activity that's in the visual cortex and all of the other activity that's just due to looking at something that's word-like. And traditionally, how we think of the fMRI is to look at the functions of the brain. So what part of the brain is active when we're performing a particular perceptual or cognitive task? What functions does it play? But it also provides other valuable psychological information. So he gives a couple examples here of when people take a placebo, the regions that are responsible for experiencing pain decrease their response. So one of the things we've learned is that the placebo isn't just, and I'm going to put just a psychological effect, right? As we get to this mind-body problem, it is a change in brain activity. Also, we can predict what people are imagining, or we can predict their kind of intentions. And I say this that we can do this with that enthusiastic cooperation. And I'm going to go show an example with pictures. Okay, so here's an example, and it's a really simplistic example of what people are actually looking at, but we can look at imagining a particular scene, and this can get much more complicated. So in this one, the control stimulus, we're showing them these bars of different orientation, and we're looking at the stimulus. In this case, they're looking at a particular oblique line, and we are measuring from 500 voxels. Okay, so these are like pixels in the brain, but three-dimensional, right? They're voxels in the brain. 500 voxels from the visual cortex, and we're seeing a certain pattern of activity as we're showing them different levels of oblique. The example down in B, the stimulus is horizontal. We can do vertical and all around. So we're looking at the pattern of activity to these different stimuli, and then we can, after a number of trials, many trials of them, again, enthusiastic cooperation of them looking at these, we can put up a stimulus and we can make a prediction. We can say, oh, now you're looking at horizontal lines. And they'll say, yes, I'm looking at horizontal lines based on that pattern of activity in that area of the visual cortex. And while this is a really simple grading pattern, I've seen where they've done this with much more complicated stimuli of pictures of mountains and so naturalistic pictures, but we can use this to make predictions about what people are seeing, imaging, intentions, and thinking about. Again, with their enthusiastic cooperation, they're running through many trials where they're telling us, this is what I'm experiencing. In this section I have called correlating brain activity with behavior. If you notice in the original outline, when I have the outline up, it's called the structure of the living human brain. I'm talking about the same thing here, right? So when we're looking at the structure of a living human brain and we're asking, what can this person do? What can they not do? What is their behavior? What is their personality? Maybe we are asking what is the correlation between that brain anatomy with their behavior. And this really all started back in the early 1800s with phrenology. It was actually a couple hundred years before that, maybe almost 200 years before that, when it was first proposed that potentially different functions of the brain different functions were occurring in different regions of the brain. But for a long time, there weren't a lot of proponents of that theory. Obviously, that's what we know occurs now. And by the 1800s, the scientists were starting to accept that, and phrenology came about. I have seen several really neat phrenological maps and models in people's offices, where what they would do, and what this is showing with these numbers on the brain, is that they would feel the bumps and protrusions at the skull, and then make some assumption about brain activity, or what that means as far as your personality, behavior, your ability with language, all of those things. Of course, we now know that phrenology is not an answer to how to look at brain activity, that at the level of the skull, that's not showing us what's happening in just feeling the skull with your fingers. That's not going to tell us what's happening in the brain. But this was around for a while, was considered scientific for a while, and then eventually it started going into more like carnival shows and so forth, as it lost more and more credibility over lost credibility over time. I keep thinking of one more thing I want to say after I've left a slide, but I used to sit in on a psychophysiology training grant talk every Monday for years. And Manny Donchin, if you get a chance to read anything by him, he was kind of one of the founders of a lot of this, some of this field. He was back when they were doing a lot of PET studies. And when I was in graduate school, the fMRI was becoming more common. It wasn't like it is now, but it was becoming more common. And so Illinois had an fMRI machine, and we're doing studies on the fMRI. And he would sometimes in that training grant talk meeting, he would talk about how we're the phrenologists of our time, that we are still making guesses, and we are still looking at correlations and making assumptions from those correlations. And we really still need the cognitive, or if I'm looking at something about cognition, I need to look at the tasks and the behavior as well as the brain anatomy, and I need to realize that I am making some assumptions there. But anyway, so we've moved on from phrenology, and for a while we used, and we still use, for people especially who cannot go in an MRI machine, if we're looking at, if we have a patient and we want to look at brain activity or brain structure, a lot of people have a pacemaker or some kind of piercing that's in a spot where something, or metal in their body somehow, so that they cannot go in an MRI. So we still do use PET for certain things. We still do use this CT scans. So computerized axial tomography, we're looking at structure, and this is basically getting X-rays, but from many different angles. So they have an X-ray beam, an X-ray detector, and they rotate. For us, because we're looking at the brain, they rotate around the head, taking X-rays from many angles. And so then they assemble this series of those successive slices that are happening over time as they're moving those machines to construct images of the entire brain, and it lets us look at the structure of the brain. This is the least expensive and most widely used method for looking at brain structure. Okay, so here is, I'm showing you some CAT scans of a woman with left hemineglect. Okay, so you have to kind of imagine, let's see if I can do this right. My lefts and rights, because of my bit of cerebellar problems, are difficult for me. On the picture, right is to your left of the person. So you kind of have to imagine spinning your head around and starting, and then starting down. You can see the sockets of the eyes, and then in one, and then moving up right through the brain, and things look pretty normal. As we move into, I'm going to go ahead and move to 4. You can start to see on the right hand side of the person, our left-hand side, this little white space there in the parietal lobes. Remember, we talked about left hemineglect being that right parietal lobe damage that causes an inability to really pay attention to the left side of space, or the left side of the body, and so forth. And then we're moving up, so we see that little tiny white space, which is the damaged brain, and then we move up the next successive slice, and you can really see in number 5 that area that is damaged in the parietal lobes. And so that gives the CAT scan gives us a pretty good idea of where there is brain damage, or where there's a tumor, and so forth, about brain structure. Okay, so we're finally to magnetic resonance imaging, which most of us have had some experience with. If you haven't had an MRI yourself, you know somebody, most likely, who has had an MRI. And this is how they're looking at the structure of whatever. Sometimes someone's getting their leg looked at, or something in their abdomen or whatever. But we're going to look at the brain, of course, because we're talking about the brain. When we're in the magnetic resonance imagery, we have this magnetic field. We're using those radio waves and the computerized enhancement to interpret that information and give us an idea of the structure. And what's happening in that magnet, so atoms with odd numbered atomic weights have a certain axis of rotation, and so different structures in our body have different amounts of water. And the MRI applies this powerful magnetic field to align all the axes of rotation of those atoms. And then they pass this radio frequency field. They pass it through, and then it stops. They start it and then it stops. And that radio frequency field tilts those atoms. And then when it's turned off, those atoms, those atomic nuclei relax, and they return to their original axes, releasing electromagnetic energy. So the MRI then measures that energy that's being emitted and tells us about the different structures. Tells us about the different the different structures because of the different amounts of odd numbered atoms, or the different amounts of water really in those structures. So the MRI provides much better imaging of brain structures than CAT scans, and we get these three-dimensional images of the brain with remarkably high resolution. And you can see a scientist there looking at an MRI. It's a little bit smaller than I'd like, but you can see just how the spatial resolution on those brain structures. You can look at anatomical details smaller than 1 millimeter in diameter. And so finally, I've put in a table from another textbook. I know some people really like to have tables and where they can see everything all together. This has very brief descriptions, not quite what I've gone into, of I think everything we have talked about. If memory serves, I'm thinking we went through all of these. Plus, she adds, this is from Laura Freeberg, she has the gene knockout. We did not talk about gene knockouts where they are any kind of genetic changes where they might have some mutation where a gene does not get expressed in some animal. Okay, so this is the summary really of research methods. You guys can pause it and look at this. I'm going to move on. Yay, we're not too far behind into genetics today.